Today we're going to be talking about Chapter 13 in your McCurnin's book, which is Clinical Chemistry, Serology, and Urinalysis. And we're going to go over this pretty briefly in an introductory manner so you have a basic idea of what we're looking for, why we're looking for it. Um, but we're going to go into this a little bit more as we go through anatomy and physiology later on in this program. So I just want you to basically understand the key terms, talk about why we do clinical chemistry. Um, we're not going to really talk a lot about chemical analyzers that we use in veterinary and human medicine, but a basic understanding of that. Also, serologic testing and urinalysis. So clinical chemistry is used to screen for disease, to assess risk before surgery, to distinguish between differential diagnoses. Differential diagnoses are the possibilities of why an animal is sick. Also, if we know that an animal does have an existing disease, we can assess the severity of that disease, monitor progression or response to therapy, which means we're sampling from the pet serially or every day for a week or every week for a month or every month or every 3 times a year. But that is serial sampling, see how they're progressing. And we also want to use it to identify patterns that suggest dysfunction. Sometimes we're taking blood on an animal that seems otherwise healthy. There's no sign that there's anything wrong with this animal. But then we start to notice patterns, and these patterns indicate that there actually is something going on, but at this point they're able to hide it or mask the disease. So what are some things that we can do that will change the analysis and make us think that there's no problem when there actually is a problem, or that there is a problem that we cannot find, or a problem that we create by the way that we sample the blood for the animal or conduct the test. So some factors that can affect the quality of the test before we get it. This is called pre-analytic factors. So things that we do prior to putting this through the machine. Our collection procedure. If we are not using the largest needle possible to get the sample, what we're ending up doing is creating more damage actually to the cells than is necessary. When we are collecting samples for a number of different tests, we're often going to be collecting it in a number of different tubes. We talked last lecture about taking a sample for a CBC in a lavender top tubes. These lavender top tubes or green top tubes have chemicals in them. Lithium heparinized, lithium heparin is in a green top tube. Calcium EDTA is in a purple top tube. And if we're putting it in those tubes first and we're mixing it, then those chemicals can get on the needle. And then when we put it into a different tube, that chemical is still in there, and that can change the sample or the test that we get from the sample. So we do recommend that you fill the red top tube first, and or if you're filling another tube that you don't invert it so that you get a chemical on the needle. You want to quickly remove any heparinized plasma or serum from clots. So if you have something that does have heparin in it, we want to take it off that clot once we've spun it down as quickly as possible. If we don't do that, the chemicals that are within the red blood cells will eventually leach into the chemicals in the plasma and change those chemicals and give us an abnormal result. So we always want to test just the serum or the plasma if we've tried to separate those two things. We do want to store these things carefully capped. We don't want to leave them open to the air because that will change things. And we don't need to refrigerate them. We want to look for anything that might interfere in the chemical machine. So if there's anything in the sample that can cause a problem. Anytime we're shipping these samples, we want to make sure that they are labeled correctly, that they're with the right paperwork, that we're handling them correctly, and that we're being shipped without delay. There will be some patient variables that will change the way that we might ship something or test something, and there may be some equipment difficulties that we need to adjust before we do any analysis. within the analytical machine, so within the machine itself, we need to make sure that these biochemical analyzers are working, functioning normally, that the analyzer has all of the pieces and parts of it that it needs, and that they are functioning within limits. Anything that we're using to prepare our sample needs to be functioning. So a centrifuge needs to be functioning normally. We need to make sure that when we are doing certain tests that we're following the directions. We need to make sure that our machine um, is matching our quality control, so that when we do controls in the morning, or before testing, that we are making sure that they are within the limits of what is normal for that machine. We need to make sure that we're operating the machine according to the instructions, and that we're maintaining the machine according to the instructions. These machines are anywhere between $5,000 and $22,000. They're really can be very expensive and almost impossible in some cases to replace. So we need to make sure that you understand proper maintenance and control. Quality controls are really important to do every single day, and if you're getting incorrect readings on your quality control, that means that your results will be incorrect as well. So quality control really leads to accurate results. So we need to calibrate. In some cases, we need to calibrate machines. Not all machines need to be calibrated, but about every 6 months or so, we want to do our calibration of the machine. We want to do routine maintenance, which includes cleaning, running daily controls. Daily controls tells us for each test if we are within the normal limits. And if you look at this Levy-Jennings chart, you can see that we're marking down where the phosphorus is testing with our control sample, and we will get a control sample sent to us now and again, especially with IDEX tested machines. They will send us a control sample. Heska as well does controls. Abaxis does not. And they will send us a sample, and we will test that sample, that known sample that has been tested on the perfect machine, and we will make sure that our machine also tests it at the right level. And if we're seeing that we have it reaching outside of those levels, that means we know that our machine is not working perfectly. So it's going to vary from day to day, depending on how much you mix it, what the variables are, how long the control has sat out for 5 minutes or 6 minutes, because all those things can matter. And if you're starting to see the change in the control, that means your machine is off. So we're going to want to do whatever quality control for that machine that is required. We may have to do blanking methods to counteract interfering substances. So that's kind of like whiting things out and starting over. We have machines that need reagents, which means these are chemicals that react with the blood or the sample that we're putting in there. And if we have deterioration of these chemical agents, that's going to cause changes within the results of the sample. Our Heska blood machine, for instance, has 3 different reagents. One's to clean, one's a neutralizer, and one is the actual reagent that works with the blood sample. And those things, we go through them very quickly. The problem is the machine can sit there for months and months and months and not be used, and those reagents will still deteriorate. So we have to replace them whether we use them or not. Want to make sure that we're monitoring the condition of our analyzer. So to do that, we want to keep maintenance records and have very standard operating procedures. If you walk into a clinic and you note that they don't have quality control for their machines, and they don't have a standard operating procedure, and nobody has taken the accountability and responsibility to take care of that machine, that is one area where if you feel comfortable stepping in, I would highly recommend that you do so. Because a lot of people say, oh, it's a machine and it's just going to run the way the machines run until it breaks down, and then we know we have to replace it. It's just not the case. We need to make sure that every single day it's giving us a correct result. Serology is a little bit different from chemistry. So with chemical testing, we're testing the chemicals in the blood to determine how the organs disease, kidney disease, pancreatitis. We can find those things by detecting the chemicals in the blood. When we talk about serology, we are testing the serum for antigens. Antigens are the little things that live on viruses or bacteria. They're on the surface of the cell that indicate to other cells what it is. Antigens are when we are looking for certain diseases. So when we have antigens, if we have diabetes, if we have some changes in hormones like endocrinology testing, if we have, we can use them for drug testing, we can use it for detecting infectious agents, and that's probably the most common one we'll use in veterinary medicine. When we're doing serology, a lot of times what we're looking for are titers. Titers are when you're looking at for concentrations of the antigen. So we can do ELISA testing, which is an enzyme-linked immunoabsorbent assay, to screen for a disease. So those ELISA tests are the ones that you will see that are one line that shows it's positive or negative, or a dot, a different colored dot that says it's normal, or there's a positive test for, say, Lyme disease or heartworm disease. Those are ELISA tests, and those can screen for certain diseases. When we're looking at titers, what we're looking at is taking serial samples, so anywhere from 1 to 3 weeks apart. The first one would tell us the certain concentration of antigen that they have in that blood sample, and then a couple of weeks later we may take another sample and see if it's increasing or decreasing, and that just tells us do we have an active infection or something that is decreasing over time. Or is this antigen staying steady, which will tell us that this is something that the animal has, maybe as a vaccine, something like that. So we're looking at the change over time. When we're doing these tests, we can do it as an ELISA test, as a radioimmunoassay test, as an immunoradiometric assay, Western analysis, an agar gel immunodiffusion, virus neutralization, hemagglutination inhibition, and complement fixation. So these are a lot of different techniques. A lot of these are sent away to a laboratory where they're trained to do these specific techniques to look for specific diseases or doing drug testing, endocrinology testing, etc. The one test, like I said, that you would do in-house in the clinic, they make it very easy for you to do, is the ELISA test, the enzyme-linked immunoabsorbent assay. And basically what this is doing is it's taking the sample, which is the blood, that has potentially the antigen for a disease in it, and then using known antibodies, so a sample of a reagent or blood that has those antibodies against that disease. The antigen and the antibody combine, and if you color the antibody and indicate that when it combines with the antigen that it will change color at a certain spot on the test, that's how you get a positive result. So that's what we see with a SNAP test. For instance, this is a heartworm, Lyme, and Ehrlichia test. We also have a fourth one, I believe, testing for Rocky Mountain spotted fever, and so this would have an additional dot. And this is a negative test, and what we do is we mix the blood with a reagent and put it in this well here. We allow it to travel up the paper, and when it gets to this level, we snap this down. It's called a SNAP test. We snap it down. It breaks a glass vial that puts the antigen into the sample, and if they find the antigen, if the antibody finds the antigen, then it will color the circle where that indicates what disease it is. If it doesn't find the antigen, then you just get this negative control, which just tells you that that you have a working test. Okay, so basically an ELISA test is binding antigen to antibody with a detection system. This is looking at it as a titer, using titration, and just seeing how it changes the color. So if you have a lot of it, it's going to have more color. If you have less of it, you're going to have less color. Urinalysis. When we collect urine, there are a number of different ways to collect it. When we collect urine, we want to do it about the same time we do a blood chemistry, chemical analysis of the blood, and or a complete blood count. And the reason that we're doing that is we need to compare what's in the urine to what's in the blood, because it's the whole, so we're looking at the whole body, we're looking at the whole system, and this is part of it. This is an example of a free catch. You can see that this person has a glove on their hand. That's really important. That's personal protective equipment. That they're using a sterile or very clean container to collect the urine. We usually catch this midstream, so we wait till the animal is urinating, stick it under, try to get at least 8 cc's of urine in that cup. There are other ways to do it. You can use a ladle, a clean ladle, or even sterilized ladle. And you can use a bowl, a clean or sterilized bowl. You can use something to hold like a coat hanger, something like a coat hanger that holds onto this cup, so you can stand a little further away if they're shy. So there are a number of ways to do a free catch. When we do a free catch, we have to assume that there's going to be some bacteria, maybe some other things in the urine. So it's an easy way to get urine, to get a basic idea of what's going on with it, but it can be contaminated, so we have to be careful about that. With a cystocentesis, that means that we're taking a needle and we're putting it into the abdomen, directly into the bladder, and collecting it directly from the bladder. This eliminates contamination from the outside or from going through the rest of the urinary tract. So it can tell us if we have bacteria that is living in the bladder itself. If we have a little bit of blood in there, that could be because we had a needle sticking into the bladder that can cause a little bit of bleeding. So a few red blood cells don't necessarily tell us anything. But this can be a cystocentesis is the couple of different reasons. We don't have contamination, and we can culture it to see if we do see bacteria. We can see what kind of bacteria there is. Catheterization is where we take a small tube and we place it into the urethra, into the vulva for a female, or into the penis for a male, and we stick it in there. It's usually typically a longer urethra, especially for a male, and we get it directly into the bladder. Now we can contaminate the bladder by doing this. So if we have an infection anywhere along the urethra, we can actually cause that infection to go into the bladder. So that can be a problem. If we are too rough with it, we can cause some damage to the urethra, so we have to be careful with that. So we want to make it as clean and easy procedure as possible. It is one way to get a urinalysis, and also we use catheterization for surgical procedures or for making sure we have a urethra that can pass urine. So there are a lot of different reasons we might use it. We can, if need be, collect urine from the cage floor or from a litter box that does not have absorbent crystals in it. So a litter box with some plastic beads in it is one way to do it. Again, we're going to have contamination, but we can look at some basic things from that urine and get a basic idea. So often we want to collect it as much as possible, as cleanly as possible. The examination of the urinalysis should happen within 30 minutes of collection. We want to keep it in cool storage. If we can't look at it right away, you can keep it in cool storage, which means the refrigerator, up to 6 hours at the best. If you have to go as long as 24 hours, you can do that, but certainly no longer than 24 hours. The sooner you look at it, the less changes are actually occurring in the urine. When we analyze urine specimen, we're going to need a couple things of equipment, and these are urine collection centrifuge tubes. So these are conical centrifuge tubes. You can see that they have a shape at the bottom that allows sediment to collect there. So it's important when we first look at urine that we look at the color. Color doesn't always mean that it's super concentrated. So the more yellow it is doesn't necessarily mean that it's concentrated. We have to look at a refractometer to determine the concentration of the urine. And we refer to it as urine-specific gravity. Urine specific gravity is how many solids are within the urine, which tells us how concentrated it is. The kidney's job is to dilute or to concentrate the urine. So if the urine is diluted, the kidney is working to some extent. If the urine is concentrated, the kidney is working to some extent. But there's a spot in the middle that we call isothenuric, which is between 1.011 and 1.015. We call this isothenuric, where the kidney is not having to concentrate or dilute. That could be normal, or it could indicate that we have a problem. Okay, so the kidney, in order to tell us that it's functioning, needs to either be concentrating or diluting the urine. So having an idea of what that concentration becomes very, very important when we combine that information with blood tests. When we get a sample, this is just plain old urine. The first thing we're gonna do is take a look at it. What is the color? Is there anything in there that we can see? Is it red? Is it super yellow? Is there stuff floating around it? So that's our direct examination. The next thing we're going to do is a chemical analysis. This is your chemical strip. This strip is very, very sensitive to light. So once you get a stick out, you want to test it right away, because if you leave it out for a very long period of time, these chemicals that are on these little pads here will change, and so that will change your result. So this is a black container. That should tell you that these are very sensitive to light. So we're going to do a chemical strip, and this is going to give us an idea as to protein, if there's any blood cells, white cells, pH of the urine. It will tell you specific gravity. There are certain things on here that I don't believe, like the urine specific gravity, I would rather get from a refractometer. Sometimes it will show white cells when there actually aren't white cells, and so I don't believe that completely. Sometimes that shows white cells when there aren't, because it's had too much light coming to it. So we kind of take those things, mark it down, and then look at the other parts and put the whole picture together. So we've got our urine specific gravity, and so we're measuring the concentration. We've done our chemical stick. The rest of it we're going to centrifuge down, and if you look at the bottom of some of these, you can see there's sediment at the bottom. So these are urine that has been centrifuged. We are going to pour that extra urine out, and what's left in that bottom will stay there. So we pour it out. It can be kind of scary to do because you've just collected it, but you pour that out. We don't want that. We put a couple drops of urine sediment stain in there, mix it up, put a drop on a slide, and look at under microscope. So super easy test to do, and we're going to be looking at some cells underneath. So gross examination, looking at color, turbidity, specific gravity, number, which is the number and molecular weight of the particles in the urine, and this indicates, as I said, renal function. The chemical evaluation of the urine, this is something that you buy from, it should be in your inventory. Reagent strip chemistries often test for urine pH, protein, glucose, ketones, blood or hemoglobin, and bilirubin. So these can be really important. The microscopic examination, we're going to look at sediment, do that sediment preparation. Make sure your microscope has been adjusted and that you're going through the fields and counting. So you're looking for cells. You shouldn't see a lot of cells or really any cells in the urine. You're looking for casts. Casts are basically what is left in the tubules, the teeny tiny little tubules that are in the kidney. If you have a concentration event and then you have a flushing event, it's kind of like Play-Doh has been trapped in a tube, and you put it through the tube and you get this this cast comes out of it. And you have to look at the cast to see, is it made up of protein or cells or debris? But it can often mean that we have something that has gone on within the kidney. So we're looking for those casts. And then we're looking for crystals. Crystals are formed when you have certain elements within the urine, like magnesium or phosphorus or calcium, and so they form little crystals, and those little crystals can eventually form into stones. So we have to be very careful. The crystals themselves make the urine very uncomfortable. When the animal urinates, they actually can feel discomfort associated with those crystals. And then we're also looking for microorganisms. So bacteria or even parasites may be present in the sample. We have to remember what type of sampling method that we used in order to understand why we might have more microorganisms or how many microorganisms we could expect. These are pictures of crystals. Oxalate crystals and struvite crystals are the most common ones that we will see with animals having issues with the urine. That's a brief overview of the chemistry, serology, and urinalysis. We'll certainly discuss some questions. If you have anything any stories to add to give a little color to the class, certainly take those when we discuss this in class.